Planetterrian Daily, episode 98 for June 22nd, 2026. Here's what's new in science, health, and longevity research. Accelerated biological aging links to higher early-onset cancer risk in younger adults via organ-specific proteomic clocks. Analyses of 2 population cohorts use biological aging metrics and organ proteomics clocks to examine generational differences. Younger adults showed earlier systemic and organ-specific aging compared with older adults born decades earlier. This pattern associated with increased risk of early-onset cancers. The work builds on tracked questions in aging biology around reliable biomarkers by linking proteomic signatures directly to cancer incidence. Follow-up studies will test whether interventions that slow organ clocks reduce those risks. The source is Nature Medicine. Now we turn from broad population aging patterns to how specific dietary fats influence cellular metabolic aging at the individual level. Laboratory comparisons showed palmitic acid triggers inflammation, toxic lipid accumulation, and cellular stress that impair insulin signaling. Oleic acid, the main monounsaturated fat in olive oil, supported insulin function and offset some palmitic acid effects. The differential outcomes trace to distinct impacts on mitochondrial and endoplasmic reticulum stress pathways. Dietary fat composition therefore influences metabolic outcomes beyond total calorie intake. The source is Science Daily. Most people assume any saturated fat is uniformly harmful, while unsaturated fats are broadly protective. In reality, the 2 saturated and monounsaturated fats tested in recent metabolic studies trigger opposite cascades inside cells. Right now, as you digest your next meal, palmitic acid from common processed foods can activate inflammatory pathways and overload mitochondria, whereas oleic acid stabilizes membranes and supports insulin signaling. One memorable distinction is that oleic acid measurably offsets palmitic acid damage in the same cell models rather than simply acting in parallel. The practical takeaway is to shift everyday fat sources toward olive oil, avocados, and nuts while trimming repeated intake of palm oil-heavy packaged foods. Future nutrition studies will track whether this swap alters long-term glucose control more than total fat reduction alone. Next, we move from metabolic cell stress to how researchers are now reprogramming cells across species with very different stress protection systems. Asian elephant fibroblasts were converted into induced pluripotent stem cells despite expanded TP53 and ILIF retrogene copies that enforce stringent tumor suppression. The protocol navigated these barriers without compromising genomic stability. The resulting lines provide a platform for elephant disease modeling and conservation genetics. The work also clarifies how tumor suppressor architecture limits cellular plasticity across species. The first derivation of elephant-induced pluripotent stem cells shows that expanded TP53 and LIF retrogene copies create a high barrier to reprogramming, yet can be navigated. The resulting lines open direct testing of elephant-specific disease mechanisms and potential conservation applications. Because the cells retain their native tumor suppressor architecture, they also let researchers map where anti-cancer safeguards begin to block tissue renewal. The source is Nature Methods. Now we shift from species-specific cellular plasticity to plans that could improve human nutrition across climates. Rutgers University researchers completed 2 complementary studies on Amaranthus species published in Hort Science. They combined morphological characterization with measurements of horticultural performance and nutrient content. The crop showed rapid growth, strong environmental adaptability, and elevated levels of key vitamins and minerals. Results position amaranth as a candidate for expanded leafy green production in varied climates. The source is phys.org. Brookhaven National Laboratory achieved an early milestone in radiofrequency control systems for the electron-ion collider. The system will steer electron-ion collisions to probe how matter's building blocks stay together. Engineers completed the first real-world validation of the controls under operational conditions. The collider aims to deliver higher precision data than current facilities on quark-gluon structure. The source is phys.org. Researchers engineered magnet-guided algae-based microbots to carry chemotherapy agents. The devices increased drug penetration into bladder cancer tumors compared with passive diffusion. Magnetic steering allowed precise navigation within the bladder cavity. The approach targets a common limitation in treating superficial bladder cancers, where drug contact time is short. The source is phys.org. A study of 17 tyrannosaur fossils revised earlier growth estimates for Tyrannosaurus rex. The predator took approximately 40 years to attain its adult mass of roughly 8 tons. The extended timeline adds 15 years to previous projections derived from smaller sample sets. Bone histology provided the incremental growth data across multiple individuals. The source is Science Daily. Researchers documented the abundance and activity of giant viruses in Arctic and Antarctic microbial communities. These viruses influence microbial population turnover and the cycling of carbon and nutrients. Their genomes encode genes that modulate host metabolism during infection. The findings expand understanding of how viruses structure food webs in extreme environments. The source is phys.org. A new refractive framework called HiSil pairs index-matched solid and liquid components with standard air objectives. The system achieves sub-micrometer resolution across centimeter-scale samples in various immersion media. It removes the working distance and cost constraints of traditional immersion lenses. Applications include intact tissue volumes previously inaccessible to high-resolution light microscopy. Nationwide cohort data indicate that switching to electronic cigarettes after smoking cessation lessens the expected drop in lung cancer incidence and mortality. The attenuation appears in real-world follow-up rather than controlled trials. Authors note that additional longitudinal evidence is required before firm clinical guidance can be issued. The finding concerns residual risk pathways that persist beyond nicotine replacement. A reported case used artificial intelligence analysis of cardiac imaging to identify disease progression that standard reads missed. The enhanced diagnostic pathway led directly to transplant listing and successful surgery. The example illustrates how algorithmic review of serial scans can surface subtle functional decline. Further validation across larger patient series is underway. A new platform combines light sheet imaging with computer-guided milling to sample defined regions inside large tissue volumes. It delivers high-depth proteomic and transcriptomic data while preserving spatial context. The method extends three-dimensional molecular cartography beyond thin section limits. Researchers expect applications in developmental biology and tumor microenvironment studies. The source is Nature Methods. A miniaturized, cloud-operated microscope enables continuous multi-contrast imaging of brain function in freely behaving animals. The device captures neuronal firing alongside hemodynamic signals for at least 24 consecutive hours. Remote access allows simultaneous monitoring across laboratories. The system targets longitudinal studies of seizures, tumors, and other chronic CNS conditions. The source is Nature Methods. Systematic proteomics of rat lungs across age, sex, and anatomical regions identified matrix maturation as the dominant aging signature. Aged tissue accumulated mature collagen crosslinks and showed reduced biosynthetic activity. Metabolic stress and immune activation markers rose in parallel. Changes were more pronounced in females and varied across lung compartments. The source is BioRICE. The SXDPAE gene sensor produces a greater than 100-fold fluorescence increase upon binding polyphosphate over ADP or ATP. It quantifies poly-P in microplate assays, stains gels, and labels intracellular granules in live cells. Stimulated emission depletion microscopy resolved subdiffraction poly-P clusters. The probe enables long-term tracking of polyphosphate dynamics in multiple experimental formats. The source is BioRxiv. Keep an eye on follow-up work testing whether slowing organ clocks through lifestyle or other means reduces early-onset cancer incidence in the cohorts already under study. That covers today's science and health news. Share this with someone who's curious about the latest research. I'm Patrick in Vancouver. See you next time. And before you go, this show is part of the NARA Network, a family of daily podcasts covering tech, science, markets, and more. If you enjoyed today's episode, give OmniView a listen, a balanced daily world news briefing that takes every side seriously. You can explore the whole lineup at neuranetwork.com. This episode used AI voice synthesis of my voice. Editorial selection and analysis are my own.